Hello, my name is Melanie Martin. I'm a professor of physics at the University of Winnipeg, and today I'm going to be telling you about tools I'm building to see inside the brain beyond the human eye. The total cost of neurological conditions is estimated to be $8.8 billion in Canada in 2000 to 2001. More than 9% of acute care hospitalizations and 19% of patient days in acute care hospitals in Canada in 2004 to 2005 were for patients with neurological conditions. 20% of patients receiving inpatient rehabilitation in 2005 to 2006 had either head injury, multiple sclerosis, Parkinson's disease, spinal cord injury, or stroke. 50% of complex continuing care stays and 65% of complex continuing care days were for people with Alzheimer's disease, ALS, cerebral palsy, epilepsy, head injury, multiple sclerosis, Parkinson's disease, or stroke. In my opinion, the major issue slowing the progress of finding effective treatments and possible cures for all these neurological conditions is the lack of ability to see changes in the brain. These changes are devastating, they cause all sorts of problems, and we can only do so much to fix them. If we can visualize and evaluate the changes in the brain, we could see the effects of treatment. We could know sooner if the treatments are working. We could know sooner if the person has the disease and if we should start treatment. For instance, with stroke. What is a stroke? A stroke happens when blood stops flowing to any part of your brain, causing damage to brain cells. More than 62,000 strokes occur in Canada each year. More women die of stroke than men, and women tend to have worse outcomes than men. Approximately 405,000 people in Canada are living with the effects of stroke. Before 1990, it took about 8 hours to see a stroke in the brain. In 1990, Dr. Mike Mosley discovered that diffusion can see a stroke as it happens. In the past 30 years since his discovery, stroke outcomes have improved. And it's happened because the diagnosis can happen sooner. Even 8 hours sooner has made a drastic improvement in outcomes. But there's still more work that needs to be done. We need to find better treatments. We need to get better outcomes. We saw the numbers on the previous slides. We need to fix that. And I believe this might come through a better understanding of the changes that we see in the images. Multiple sclerosis is an autoimmune disease of the central nervous system. What that means is the body thinks your own brain is something that shouldn't be there, and the body will attack it. Canada has one of the highest rates of MS in the world, and it is estimated that 77,000 Canadians are living with multiple sclerosis. MRI is really nice. It can detect many lesions in the brain due to MS. Unfortunately, there appears to be no correlation between the number and severity of lesions detected with MRI and the health of the person being imaged. This has caused some people to think that MRI shouldn't even be used in the diagnosis and monitoring of treatments of MS. A European study suggested that it might be possible that an image taken today can indicate how a patient might feel in 10 years. We need to do better than that. We need to find the correlation between the image we see today and how the patient feels today, or the person feels today. My theory is that with MRI, in its current state, we cannot distinguish between the types of lesions in the brain. We might see lesions from demyelination. We might see lesions from inflammation or edema, and we are unable to distinguish between these types of lesions. I believe that if we knew what types of lesions we had, we knew the numbers of lesions we had, we knew where the lesions were, we might be able to get a better correlation with the health of the person. If we could make this correlation, we could do a better job at planning and evaluating treatments. Dementia is a general term for loss of memory, language, problem solving, and other thinking abilities that are severe enough to interfere with daily life. Thankfully, it's not me forgetting my keys where I put them. It's me forgetting what my keys are used for. Alzheimer's disease is the most common cause of dementia. More than 747,000 Canadians are living with dementia. A diagnosis of Alzheimer's disease changes the life of the person with the disease, and also it changes the lives of their friends and family. The friends and family become caregivers for these people. They have to provide a lot of help for the person with Alzheimer's disease, and eventually, inevitably, it leads to the person with Alzheimer's disease moving into a care home. Unlike multiple sclerosis, we are just now starting to see early anatomical changes in the brain due to Alzheimer's disease with MRI. We still need to correlate these changes with symptoms of the disease. PET, positron emission tomography, that kind of imaging can be used to detect functional differences in brains with dementia. For instance, glucose uptake is different between brains of people with different forms of dementia and brains of people without dementia. As of yet, imaging does not provide enough information to be routinely used in diagnosing Alzheimer's disease. I believe that we can do more with imaging to understand Alzheimer's disease and to make it a more useful tool. Schizophrenia is a serious mental disorder. A common early symptom of schizophrenia is psychosis, or the experience of loss of contact with reality, which usually involves hallucinations and delusions. 300,000 Canadians are affected by schizophrenia. Diagnosis of schizophrenia is difficult and time-consuming. On autopsy, brains of people with schizophrenia appear to have less densely packed fibers than brains of people without schizophrenia. Without being able to measure the density of axons and fibers in a living brain, it's difficult to determine if what they're finding on autopsy is actually true. Is that a characteristic of schizophrenia? Are the fibers more densely packed in the brains? Are the fibers less densely packed in the brains of people with schizophrenia and not in the brains of people without schizophrenia? Is this less dense packing a cause of schizophrenia or a consequence? For instance, if people who later on in their lives go on to develop schizophrenia, are they born with less densely packed fibers? Or does the density of fibers decrease later in life? If we knew that, we could have an earlier diagnosis, and we could have another target for treatment. Autism spectrum disorder is a neurodevelopmental disorder that impacts the brain's development. The result of the result of autism spectrum disorder is that most individuals experience communication problems, difficulty with social interactions, and a tendency to repeat specific patterns of behavior. An estimated 1 in 66 people between the ages of 5 and 17 have been diagnosed with an autism spectrum disorder. When I give talks to public audience across mostly North America, but across the world as well, I'm told that some people believe that the brains of people with autism might have more densely packed fibers than the brain of people without autism. Just like schizophrenia, if we could measure the density of fibers in the brains of a living person, we could find out if this was true. Is this actually a characteristic of autism spectrum disorder? And if it is, are people with autism spectrum disorder born with more densely packed fibers, or does that develop later in life? This could help us with some treatments, potentially. So what am I doing? I am developing techniques to see inside the brain. I'm trying to get information out of the images to understand the structure of the living brain. I'm doing this with MRI, and this means that I can find this information non-invasively. I don't have to take a biopsy of your brain. I can just take a picture of it. I'm using diffusion MRI for the most part. That was that technique that Michael Mosley used to see stroke early. What it does is it looks at the movement of water molecules in the brain. And from that movement, we can determine the size and density of axons in the brain. This could help with schizophrenia and maybe even autism spectrum disorder. We saw we can detect stroke early. We need to understand why and what effect that has and what treatments we can make. It appears that the combination of diffusion MRI with other forms of MRI can help to differentiate lesion types in MS, multiple sclerosis. And we've seen that with some of the images that we've taken. So hopefully with that information, we can start to correlate how a person's brain looks today with how they feel today. We have also used diffusion MRI to measure properties of small regions in the brain, which were previously too hard to distinguish. And from that, we are starting to find changes in the brain of Alzheimer's disease due to genetic differences. So I hope I've shown you that detecting small changes within the brain could potentially lead to faster and easier diagnosis, a better means to evaluate treatments, and a better means to plan treatments. Thank you.
Hello, Soapbox Canada. I'm Dr. Sara Victoria Good, and I'm a professor at the University of Winnipeg in Winnipeg and also adjunct to the University of Manitoba. I'd like to talk to you today about some of the work that we do in my lab using evolutionary epidemiological approaches to study the function of insulin superfamily genes in metabolic disorders such as type 1 diabetes. I use integrative approaches in the lab. We use a variety of techniques in the lab. As a geneticist, we use a variety of different machines and tools, but I also do a lot of bioinformatics analysis, so a computer is a fundamental part of research in our lab. And we also apply a variety of different statistical models to our data. So individual strengths and statistics are also useful. And we use animal models such as mice and fish. So a fundamental question in genetic epidemiology is, why do individuals differ in their susceptibility to disease, such as type 1 diabetes? And you may look to systemic answers for this. So why do tissues dysfunction? Why does the pancreas start to break down, for example? Or you may dig a little bit deeper and say, well, what are the cellular processes that lead the pancreas to stop producing insulin in sufficient quantities? And, or perhaps even deeper, to look for genetic causes. What are there different genetic variants in the genomes that confer an increased disease risk? And lastly, you may even ask evolutionary questions, as I'm prone to do. Why or where do these variants exist in the human genome? And have there been evolutionary pressures that have shaped where these susceptibility loci lie? A major tool in genetic epidemiology has been something called the genome-wide association study, or GWAS, that directly enables you to use statistical approaches to look at the relationship between genetic variants and disease susceptibility. Another more recent approach is called the transcriptome-wide association study, something that we've employed a little bit in our lab, where you're looking at variation in gene expression, or transcriptome, and its association with disease. So looking a little bit deeper here at this cellular level. You can also, again, as I spoke about, I also use evolutionary approaches in my lab, which I don't have too much time to talk about today. But one of the take-home messages you get from this is that in order to understand something like disease, you can integrate and look at a variety of different levels, and these integrative approaches can be more powerful when you find consistency among signals in identifying the causes of disease. So as I said, I've been studying the insulin superfamily for about 10 years, and the insulin superfamilies consist of two major groups of peptides: the insulin-like growth factor, they're involved in metabolism, growth, and development, and the relaxin peptides that are involved in reproduction and neuroendocrine. I'm working mostly on insulin and its role in insulin, and I'll talk about type 1 diabetes a bit today, and also I've worked on insulin peptide 5, which I'll speak briefly about. So what is type 1 diabetes? Type 1 diabetes arises from the failure of the pancreas to produce sufficient insulin. So in a healthy person, our pancreas is secreting insulin, especially after we've had a good meal that's broken down, either a sugary drink or things that are then broken down into glucose. That glucose is circulated in our blood, which gives a signal for insulin to be released from the pancreas, and that insulin be binding with the glucose and that helps to transport the glucose into cells, which we need to make energy. But in individuals with type 1 diabetes, the pancreas isn't producing sufficient insulin, usually produces some, but not sufficient. So we end up having a lot of extra glucose in the cells, which is called hyperglycemia. So as you probably know, Canada's University of Toronto's Banting and Best were instrumental in discovering that you could synthesize, discover the peptide insulin, and that you can synthesize this and give injections of insulin to diabetic patients, which is required for individuals with type 1 diabetes. And this would help them enable to be able to take that glucose into their cells. Well, as you can imagine, many people have tried to look for whether there's a genetic basis of risk for type 1 diabetes. And indeed, this has uncovered that there is a high heritability of risk for type 1 diabetes, meaning that if someone in your family has type 1 diabetes, you have a higher risk of also having it. And this has been shed on by GWAS, genome-wide association studies, as I spoke briefly about, and she's uncovered many different genes in the human genome that are associated with this elevated risk. There's also environmental risks, things such as infections, diet, weight, stress, and even the microbiome, where that bacteria in our gut is influencing our susceptibility for acquiring type 1 diabetes, which is usually diagnosed during childhood. Well, as it turns out, the worst thing, the main thing that leads to complications in type 1 diabetes is just this prolonged exposure to hyperglycemia. So as long as individuals can keep their insulin injections done in a very regulated way to keep their blood glucose down, then the complications from diabetes are not so severe. But if there is prolonged exposure to hyperglycemia, this leads to a variety of different complications. There's problems with the eyes, with the kidneys, leg problems, feet problems, as people may well be aware of. And so of the many different things that can lead to complications from diabetes, and this includes type 1 as well as type 2 diabetes, this glycemic control is a fundamental concern. And so measuring something that's called HbA1c, which is a test that you get at the doctor's that gives you an estimate of your average blood glucose over the last 3 years. This is of relevance for public health because it's shown that this prolonged hyperglycemia can lead to micro and macroevolutionary complications that result from the accumulation of what are called advanced glycation end products. Well, what are these AGEs? Essentially, when you have this high levels of sugar in your blood for too long, the sugar moieties attach themselves to a variety of different proteins, especially proteins that are in our eyes and our kidneys, and these lead to AGEs, which then can lead to microvascular problems and lead us to having problems with our eyesight or kidney disease. So identifying the causes of genetic variation among individuals that may lead you to have a higher or lower sustained HbA1c, as it's called, or your average glucose over time may provide important insights into screening, prevention, and treatment. So as I mentioned, you can look for these genetic variants at a variety of different levels. And I performed a study in collaboration with researchers at Sick Kids Hospital to use something called a transcriptome-wide association study to look for associations of genetic factors with HbA1c in a cohort of individuals with type 1 diabetes. And I won't show you the results of all this, but essentially we end up taking genetic markers, we use statistics. Sometimes you can also add this with biological markers, and we ultimately performed analysis to identify whether variation in gene transcription or expression could be associated with these blood glucose levels. And we did identify a variety of genes, and some of these genes matched genes that were also identified from other approaches like GWAS. And so by having multiple sources of evidence that are pointing in the same direction, you essentially get better and better clues that maybe you're onto something. So in conclusion from this study, there's evidence that variation among individuals in DNA sequences that control gene expression may also influence variation in these levels of sustained blood glucose. So another group of studies I want to tell you about in the lab, something funded through my NSERC research, where we're looking at the function of insulin like peptide 5. So insulin-like peptide 5 is most highly expressed in the gut. So insulin is most highly expressed in the pancreas, but insulin-like peptide 5 is most highly expressed in the distal colon. And this is interesting because the distal colon is the part of our colon that has the highest number of bacteria. And these bacteria we know are essential for performing a variety of different functions now in metabolism. So I'm sure you've all heard lots of things about having a healthy gut. Well, and drinking your yogurt or your probiotics, taking your probiotics, and that's to sustain a good quantity of so-called healthy gut bacteria, which are essentially for performing optimal digestion. Well, for a variety of reasons, we had hypothesized that maybe insulin-like peptide 5 is also involved in helping to keep our gut microbiome healthy. Our hypothesis was essentially that insulin-like peptide 5 helps the gut not respond, so it's anti-inflammatory, to the presence of bacteria. So in doing so, it would help new bacteria colonize the gut. So we performed this experiment here at University of Winnipeg where we injected these mice with insulin-like peptide 5 and tested essentially is there evidence of an anti-inflammatory action, which would basically be saying it could be probiotic friendly. And we have some data suggesting that it is indeed, it does help to induce an anti-inflammatory response in the gut. So we measured a bunch of molecules that are involved in the immune response and looked for down regulation shown in blue or up regulation shown in red, and are developing a hypothesis that it may be anti-inflammatory and so probiotic friendly, essentially. So this is just a short summary of the research, but want to let you know, go back to my first point, which is that the approaches that we take in the lab, as biologists are increasingly taking, are integrative, where we're combining some laboratory work with lots of in silico analysis, in my case, statistical analysis, as well as work in animal models such as zebrafish and macaca, which we have here at the University of Winnipeg, as well as mice. So I'd like to thank all the wonderful students that working in my lab, as well as NSERC for many years of funding, and great collaborators at Sick Kids and the University of Manitoba. And thank you very much.
Hello. If we haven't already met, my name is Kaye. I'm a master's student, and today I want to tell you about my project. I'm working to develop diagnostic tests for RNA viruses using a technology called Toehold switches. You probably already know a little about RNA viruses. Coronaviruses, Ebola viruses, and flu viruses are all examples of RNA viruses. But this might be the first time you've heard about Toehold switches. If that's the case, let me be the first to explain them to you. To start off, I want to remind you about three things: DNA, RNA, and protein. You might remember from biology classes that DNA, RNA, and protein are related. If you imagine a factory, DNA is like your master set of plans. DNA contains all the very important instructions for your factory. And you want to make sure to keep those plans safe. That's why whenever you need to use those instructions, you make copies of the parts you need. You know, in case you spill your drink on it or something. RNA is made and used in the same way. RNA carries specific sets of instructions and can be thrown away when it's not needed anymore. The factory can read the instructions and make a product. That product is like protein. And just like how different factories can make anything from gummy bears to airplanes, proteins come in all sorts of different shapes and do all sorts of different things. Toehold switches are made of RNA. They carry specific instructions just like RNA can, and they have one more special feature. Just like a light switch, Toehold switches can be turned on and off. In my project, I'm using toehold switches to test for viruses. When the virus I'm looking for is in the sample, the toehold switch turns on and sets off a chain reaction that turns my test paper purple. I think the science behind this is really cool, so I want to explain to you how it all works. The toehold switch diagnostic test takes place on a 2mm paper disc on a 1.8 microliter drop of water. It's tiny, but this tiny paper disc is full of toehold switch RNA, all the molecular machines to read the RNA into protein, and a special yellow dye. When it's off, the toehold switch looks like this. It's twisted closed. Ribosomes, the molecular machines that read RNA into protein, can only attach here at the top of the loop. But since the toehold switch is twisted closed, they're stuck and they can't read the instructions. No protein is made and the test paper stays yellow. But look what happens when the virus is in the sample. The virus RNA latches onto the toehold switch and unravels the twist. This switches the toehold switch on. This interaction is specific, like a key or a fingerprint reader. When the toehold switch is on, the ribosome can go ahead and read the instructions to make the protein. The protein in my test changes the color of the dye from yellow to purple, and it's a process that we can even see by eye. This is exciting to me for two reasons. First, you already know that I think the mechanism behind toehold switches is really cool. Toehold switches simplify complex viral diagnostic testing into a yes or no answer. The second is that we can use toehold switch paper-based diagnostics to address a real-world need. Emerging infectious diseases are diseases that have caused, are causing, or we think will cause outbreaks. They're difficult to deal with on a public health level because the pathogens that cause emerging diseases are often so unfamiliar to us. If you think of recent epidemics like Ebola, Zika, and COVID-19, you'll realize that not only do they tend to spread quickly, but that it's often the poorest that are hit the hardest. For emerging diseases, vaccines are often unavailable, and specific therapies are under development. We control outbreaks by preventing the spread of disease to others. We rely on early detection. But traditional diagnostics are expensive. They require sophisticated infrastructure and highly trained staff and expensive reagents. Samples are sent to a central testing lab, sometimes hundreds of kilometers away. At best, the sample might be tested the next day. Often, we see labs overwhelmed during outbreaks. The flood of samples they receive is more than the resources allow them to test at any given time. Meanwhile, people who are otherwise healthy may need to quarantine until they receive their test results. But what if we could test for deadly viruses faster, cheaper, and closer to the source? By developing a paper-based Toehold switch test, we can strip away the need for complex lab instruments. We're looking for the presence or absence of a virus's genetic material. This is the same principle as the PCR, a common lab test, but we simplify so that in its final form, the Toehold switch test can be run in a local clinic at a fraction of the cost. With this new platform, we hope to make diagnostics more accessible and stop future outbreaks closer to the source.